Okay, hello everyone. My name is Brian Godwin. I'm a director of sample prep reagent development in Roche Sequencing located in Pleasanton, California. My team is primarily working on updates and improvements to current on-market target enrichment solutions, as well as working on novel target enrichment solutions, including what I'm going to be talking about today. This is HyperPETE, a novel primer extension target enrichment solution for rapid and reliable variant detection results. So with that, I will get started. So what is kappa HyperPETE? It's a novel target enrichment capture method. The capture is based on primer extension target enrichment. This is where the PETE part of the name comes from, and it uses shotgun libraries prepared from DNA or RNA as input into capture. It combines hybrid capture performance with the ease of use and turnaround time of amplicon workflows, including anchored multiplex workflows. It provides confident answers for challenging and precious samples, and it can be completed in a single-day workflow when you start from nucleic acid and through to pooled and enriched libraries. It detects all major somatic variant classes. This includes single nucleotide variants, short insertions and deletions, copy number variants, microsatellite instability or MSI, and fusion transcripts, both known in novel partners from RNA sample input. Panel sizes at launch include DNA panels of 5 kb to 300 kb and RNA panels of 5 kb to 50 kb. It includes both custom and catalog content, and it's integrated with Navify Mutation Caller, which is a cloud-based secondary data analysis solution. Finally, it provides fast custom panel ordering turnaround time for rapid deployment and testing. And the solution includes everything from extraction through to sequencing ready libraries with the integrated data analysis solution, primarily for FFPET samples in this case. So we provide the extraction solution for FFPET samples, not necessarily for the other products or the other targets. So here's a workflow overview. We start with either DNA or RNA as input. For DNA, we can use the kappa HyperPrep kit for any DNA samples that are already fragmented, and this would include cell-free DNA types of samples, or the kappa HyperPlus kit for unfragmented samples for things like FFPET DNA samples. For RNA, we use the kappa RNA HyperPrep kit, and the RNA is primarily for fusion transcript detection. Moving on, the libraries that are generated here go into our kappa HyperPETE kit. And from the start of the DNA for RNA, it's about a single-day workflow. So you'll have enriched and pooled libraries in about a one-day workflow. These libraries go into sequencing, and we're verifying and validating on both the Illumina NextSeq and Illumina MiSeq instruments. And the data coming off of the sequencer is uploaded to our Navify Mutation Caller, which again is our cloud-based secondary analysis solution. And one of the outputs are VCF files. We also provide enrichment performance metrics as well as sequencing performance in the output of the Navify Mutation Caller. So how does kappa HyperPETE technology work? So what I'm showing here denotes different library fragments that come out of our shotgun library solution. So I'm not going to go through how those library prep solutions work. I'm just going to show what comes out of that. So we have our universal dual index adapters, both with or without UMIs on these libraries. For all of the somatic applications that I'm going to talk about today, we're using UMI adapters or unique molecular index adapters for identification of unique molecules, as well as some error correction in our analysis pipeline. So in the top, you see the blue molecule, which is an on-target molecule. So the example is gene A, exon 10. You want to target that. All of the other colors are denoting off-target molecules. The first step is to put in a biotinylated primer into our reaction, and this gets extended by a DNA polymerase. Think of it as one cycle of PCR, just a primer extension. After this primer extension, we apply these to streptavidin beads where we capture all of the fragments that have that biotinylated primer extension product, and we wash away all of the off-target molecules. What I don't show in this slide is that there are still some off-target molecules that have primer extension on them. And what we've seen in development of this technology is that we have about a 5 to 15% on-target rate after this step. So one of the novel features of HyperPETE is the second release primer. So we call this our release primer hybridization and then extension. Once this gets extended, it knocks off the target molecules, the on-target molecules, off of the bead, where we can collect that in our solution, and we transfer that supernatant into a new tube where we do a PCR reaction. And this is just a universal PCR reaction using Illumina PCR primers, universal primers. Following PCR amplification, we QC, pool, sequence, and analyze those libraries. And this enrichment portion of the workflow, from the time you have a library to the time you have a pooled and QC library ready for sequencing, is about 4 to 4.5 hours. So Kappa HyperPETE has a fast turnaround time. So I'm going to show you some comparisons here. So Kappa HyperPETE is on the far right. This is a faster workflow than hybridization capture, primarily because hybridization capture technologies have an overnight hybridization reaction. It has a similar workflow time to anchored multiplex methods. And then lastly, in comparison to amplicons, although the workflow takes a little bit longer than these amplicon workflows, we process fewer samples when we're covering long contiguous regions. Panels or primers need to be split into multiple reaction tubes to capture or to enrich these long contiguous regions. So HyperPETE application turnaround time is dependent primarily on the different library prep methods. For somatic tissue DNA, we're using the Kappa Hyper Plus solution, which includes a fragmentation step. And this brings our total turnaround time of about 9 hours. For somatic cell-free DNA, we use Kappa Hyper Prep, where fragmentation is not required. So this takes a little bit less time. This is at about 8.5 hours. And so finally, we have our somatic tissue RNA prep. This takes a total of 9.5 hours. And the point here is the enrichment workflow is the same across all of the different applications. So I'm going to show some preliminary data, and this is for our catalog somatic oncology panels. I'm going to start with the somatic tissue DNA application. So what I'm showing here is our Kappa HyperPETE pan-cancer panel. This is our gene or our target list. So feel free to look through this entire list. Maybe you'll see some of your favorite genes in here. This panel covers 1,132 CDS regions across 86 oncogenes, covering single nucleotide variants, insertions and deletions, and copy number variants. It also contains 190 MSI loci for characterizing MSI status of samples, and this is denoted in purple. The capture panel size is approximately 300 kb, and this is the upper end of our panel sizes at launch. Samples used in this study include FFPET samples. There are 6 low quality, 6 mid quality, and 4 high quality normal adjacent tissue samples to assess enrichment metrics, as well as some cell line reference samples, including Seraseq lung and brain CNV mix, Horizon structural multiplex reference standard, and genomic DNA representing MSS status. So these are just standard cell line DNAs. And then there are 2 custom mutant genomic DNA mixes that are proprietary to our development team. And something that is missing here is for the FFPET samples, we have some FFPET samples with known MSI status that are included here. So experimental setup. FFPET DNA was extracted using the Kappa NGS DNA extraction kit. This is a new kit that is currently in development. And quality was measured using the Kappa NGS FFPE DNA QC kit, also a product that's in development. Libraries were generated using the Kappa Hyper Plus kit in combination with the Kappa NGS FFPE DNA polishing kit and Kappa Universal UMI adapters with either 10 nanograms of input or input based on quality assessment. And you'll see a quality assessment over here. Let me just turn on my laser pointer. So the DNA input quality input amounts range from 64 to 185 nanograms for low quality DNA, 30 to 55 nanograms for mid-quality DNA, and 22 to 25 nanograms for high-quality FFPET DNA. Libraries were captured using the Kappa HyperPETE reagent kit in the panel that I described earlier and sequenced on an Illumina NextSeq 550 instrument. And reads were subsampled to 30 million paired end reads and analyzed using Navify mutation caller to assess enrichment and variant detection performance. So over here in the chart are the results, enrichment performance results. Up on the top left are the 10 nanogram input amounts on target rate, and on the top right are quality adjusted input amounts. And what you can see is the on target rate is not impacted by input amount or quality of the input material, and our on target rates range from 69% to 78% across all of these samples. Percent panel exon bases greater than 300x and 500x was 95% across all sample qualities when using quality adjusted input amounts into library preparation. So that's what you're seeing here in this box. So it's over 95% in all cases, regardless of quality, because we've used the quality adjusted input amount to improve our performance of low quality input. Percent panel exon bases greater than 300 and 500x were variable but predictable when using strictly 10 nanograms of input. Low quality and mid quality DNA resulted in lower performance compared to higher quality DNA. So this is what you're seeing over here. Low quality FFPET samples perform with a worse overall depth of coverage compared to mid quality, and then in comparison to high quality and reference cell line DNA. So we think it's important from a customer perspective to use quality adjusted input amounts so that you get your most reliable results. As for variant detection performance, I'm giving you an overview here. So we start with short variants. This includes both SNVs and indels. And we had 99.2% of all of the variants or the short variants detected in these samples. And this is 125 out of 126 expected variants. For CNVs, we detected 9 out of 10 of the expected variants, or called 9 out of 10 of these. For one of the CNVs, we missed this call, but that's because it was called as low confidence. So you can still see the output of it. It's just not considered a high confidence call. And then MSI status was correctly assigned for the 12 MSI FFPET samples and 6 MSS cell line samples. So that's what you see here. All 6 were correctly identified as microsatellite stable, and the 12 that we know are microsatellite unstable are all called MSI high. Moving on to somatic cell-free DNA, we're using the same panel here. So there's nothing new to this target list at the top, but there are a few things to note here. So for cell-free DNA variant calling, CNV calling is not enabled. And then also MSI calling is not enabled for cell-free DNA samples in our analysis pipeline. Capture panel size is still 300 KB. And then samples used in this study. There were 3 healthy donor plasma cell-free DNA samples, and these were used to assess enrichment performance metrics through the pipeline. And then there were 3 Sera-seq ctDNA complete mutation mixes with catalog numbers there. These represent 1%, 0.5%, and 0.1%. And these are all reference cell line samples. And these were used to assess both enrichment and variant detection performance results. So similar to the last slide, starting with experiment setup, Libraries were generated using the Kappa HyperPrep kit in combination with the Kappa Universal UMI Adapters kit with either 10 nanograms, 30 nanograms, or 50 nanograms of plasma cell-free DNA or fragmented reference cell line DNA as input. Libraries were captured using the Kappa HyperPETE reagent kit and sequenced on an Illumina NextSeq 550 instrument. Reads were subsampled to 60 million paired end reads and analyzed using the Navify mutation caller to assess enrichment and variant detection performance. And here are those enrichment performance results. So on target rate was between 67 and 74% with good performance across all sample input amounts and types. So here, plasma cell-free DNA is in green and the reference cell line and cell-free cell line DNA is in blue. So you'll see on target rate is very stable and is similar across all different input amounts and types of input material. Percent panel exon bases greater than or equal to 1000x was greater than 85% across all sample types and quantities. Better performance was observed at higher input amounts and when using cell line DNA as input. So this is what I'm showing over here. At 10 nanograms, you can see a slight performance drop-off for the 1000x depth of coverage, and 30 and 50 nanograms are both higher. The other thing worth noting is plasma DNA or plasma cell-free DNA performs a little bit worse compared to the reference cell line DNA, and this is primarily due to the multimodal nature of plasma cell-free DNA and the enrichment performance of those longer the longer fragments that are in those samples. Percent panel exon bases greater than or equal to 3000x showed similar trends to the 1000x metric, but with a larger observed impact. So if we look over here at the 10 nanogram input, as expected, none of these samples are performing well at the 3000x depth metric. 30 nanograms has an improvement, and then 50 nanograms has an improvement, a continued improvement here. Something to note is that increasing read pairs used in analysis to 110 million, the percent panel exon bases greater than 3000x improved, ranging from 73% to 95% from 50 nanograms of input. This data is not shown, but this demonstrates that increasing the read depth will lead to better overall performance in this particular metric. As for variant detection performance, here is the overview. So 100% of short variants, including SNVs and indels, at 1% allele frequency were detected across all reference cell line DNA samples at all input levels. So that's what's being shown down here. And then 88 to 94% of short variants at the 0.5% allele frequency were detected across the reference cell line DNA samples. Something to note here is that variants that were not detected were present, but did not get called due to the threshold settings applied in the analysis pipeline. And one of the key features of this pipeline is you can change these thresholds depending on what you're looking to do as a researcher. Moving on to somatic tissue RNA. This is the panel. So this is our Kappa HyperPETE lung cancer fusion panel. And this is our gene and exon list. So the way to read this table is in the header is the gene name, and below that are the exons that are covered. So this panel content covers 16 oncogenes, covering 137 exons designed to detect both known and novel fusion partner transcripts at both the 5' and 3' junctions of these exons. 4 housekeeping genes serve as positive enrichment controls in the workflow, and I list those 4 housekeeping genes here. It's worth noting that these housekeeping genes, although they're expressed in many different tissues, they're at a low level. Samples used in this study, there were 4 normal adjacent FFPET RNA samples used to assess enrichment performance, and then 2 reference cell line RNA samples, including Horizon ALK RET ROS RNA fusion and Sarah-seq fusion RNA mix V4 were used to assess enrichment and variant detection performance. Experimental setup was RNA quality was determined with the DV200 score using the Agilent RNA 6000 pico assay on the bioanalyzer. Libraries were generated using the Kappa RNA HyperPrep kit in combination with the Kappa Universal UMI adapters with either 10 nanograms or 50 nanograms of RNA while adjusting PCR cycles based on the input amount and the DV200 score. Libraries were captured using the Kappa HyperPETE reagent kit in combination with the Kappa lung cancer fusion panel and sequenced on an Illumina NextSeq 550 instrument. Reads were subsampled to 540,000 paired end reads and analyzed using Navify mutation caller to assess enrichment and variant detection performance. So enrichment performance results. Following rRNA read removal, which makes up around 2% to 13% of all in all of these samples, on-target rate was 92% to 97% with good performance across all sample input amounts, qualities, and types. So that's what you're seeing here. So low quality, mid quality, high quality, as well as the reference cell line RNA all performed very similarly regardless of input amount. Housekeeping targets, which are used as the enrichment controls, as I talked about earlier, are considered on-target reads and make up between 40 and 60% of the reads, depending on the sample type. So the data is not shown here. Just remember, it's making up between 40 and 60% of all the reads in these panels. And then as far as variant detection performance, 100% of fusions in the reference cell line RNA samples were detected. So both at the 10 nanogram and the 50 nanogram input amounts. One thing that is worth noting is the EGFR Sept14 variant in the Sera-seq fusion RNA mix V4 was manually curated as the fusion caller and Navify mutation caller identified a different EGFR partner that has an homologous sequence to Sept14. So ultimately the enrichment works, we get the correct sequence, the caller just happens to be calling a different homolog. So in summary, the Kappa HyperPETE workflow and reagent kit provides a fixed and customizable content target enrichment solution for reliable detection of all major somatic variant types, including SNVs, indels, CNVs, novel fusion transcript partners, and MSI status. It's a fast and easy workflow that generates sequencer-ready libraries in a single day when starting from purified nucleic acid. It's a novel primer extension target enrichment solution that provides performance equivalent to overnight hybridization capture technologies. And it shows flexibility in nucleic acid input amounts from 10 to 50 nanograms or quality adjusted for FFPET DNA samples and sample types including high quality DNA, plasma DNA, FFPET DNA of varying qualities, and FFPET RNA of varying qualities. And with that, I would like to thank you for your time.